So this is a presentation on stem cell culture made by Maximiliano Cornejo, Ernesto Gutierrez, Gabriela Ramos, and Juan Paradame. We'll talk specifically about advances and challenges in stem cell culture. We'll divide this presentation into 5 parts, the first being the introduction, then we'll talk about two-dimensional culture techniques, then we'll talk about three-dimensional culture techniques, then some challenges that we're facing in this area, and finally a conclusion.
Stem cells, or SCs, have the capacity to self-renew and differentiate into specialized cells and are defined by their region and degree of potency. Pluripotent stem cells are capable of unlimited self-renewal and differentiation into any of the over 200 types of cells in the body. There are two sources of pluripotent stem cells. First, embryonic stem cells, or ESCs, are derived from the inner cell mass of a pre-implantation blastocyst. The pluripotency is controlled by an intrinsic regulatory network of core transcription factors, octamer binding transcription factor 4, sex-determining region YBOX2, and an analog home box. Second, the induced pluripotent stem cells are derived by the ectopic or elevated expression of 4 transcription factors, OCT4, SOX2, Croppel-like factor 4, and NYC proto-oncogene, which is essential for induction of pluripotency in somatic cells. The type of stem cells are the mesenchymal stem stromal cells, or MSCs, which are isolated from adult sources such as bone marrow and adipose tissue or perinatal tissues such as umbilical cord, cord blood, placenta, and amniotic fluid. These cells are characterized by hadron growth to plastic culture plates, exhibiting clonal growth. They are positive for mesenchymal surface markers like CD90 or CD73 and negative for hematopoietic lineage markers such as CD45 and CD34. These cells are multipotent and differentiate into only a limited cell type such as osteogenic, chondrogenic, and adipogenic cells. Their self-renewal and differentiation potential is dependent upon the source of isolation. In addition, MSCs derived from adult tissues are also affected by aging and exposure to environmental stresses, which could alter genetic stability. In comparison to adult MSCs, obtained from perinatal tissues exhibit higher growth and stemness potential. 3D culture studies have focused on the maintenance of stemness by extrinsic factors such as mechanical forces between both cells and their 3D microenvironment. They are grown in plastic culture dishes coated with ECM components such as gelatin, matrigel, or collagen, or a mouse embryonic fibroblast (MEF) feeder layer to aid in attachment. Mouse embryonic stem cells can be maintained in the pluripotent state by either culturing them on MEF feeder layers or in medium continuous leukemia inhibitory factor (LIF), while human embryonic stem cells need the LIF and the addition of fibroblast growth factor 2 and transforming growth factor beta 1 in the culture medium. There are 2 consecutive states of pluripotency in embryonic development, the early naive state followed by a primed state, from which mouse and human embryonic stem cells are derived respectively. Naive pluripotent stem cells are rarely pluripotent cells capable of differentiation into both extraembryonic and embryonic tissues, whereas primed cells are derived from a more advanced state of development and differentiate into only embryonic tissues. 2D cultures conditions favor non-specific differentiation of pluripotent stem cells. However, differentiation can be directed to specific lineages by controlling the composition and organization of the ECM growth factors and attachment substrates, which can send mechanical signals for cell fate determination. Mesenchymal stem cells are naturally adherent and do not require xenogenic substrates for attachment to culture plates, although they are typically cultured in the media containing animal serum. Mimicking the in vivo microenvironment such as hypoxia has been shown to enhance stiffness in this type of cells, resulting in a significant proliferation without the loss of multipotent differentiation. Expansion of MSCs in 2D cultures is highly inefficient. Usually, expansion consists of growth in multiple tissue culture flasks to increase the surface area, while uniform distribution, growth, and harvesting processes are important to minimize heterogeneity and improve cell yield. MSEC differentiation is achieved by using cell-specific differentiation media and assessed by translational and transcriptional gene expression as well as ECM deposition. However, expansion of MSCs in monolayers leads to phenotypic changes whereby spindle-shaped MSCs exhibit a broad and flattened morphology upon passaging, which is in turn alters cell fate and differentiation potential.
So next thing is that we're going to talk about 3D culture techniques. So in brief, these were developed to counteract the inherent problems with 2D culture, and they better represent the native microenvironment in which cells grow. So the easiest form of 3D culture is by using spheroids, which are formations of multicellular aggregates and can be formed either by hang and drop rotating culture, low addition culture plates in suspension. And there's a type of spheroid that we call embryoid bodies. These mimic the embryonic development. There's many factors that affect embryo body differentiation, like size and quantity. And the greatest challenge is that we don't use this for long-term cultures because at the center of embryo bodies, diffusion is much more difficult. So oxygen and nutrients don't generally get there, and necrosis usually shows when using long culture techniques. So certain advantages of EBs are that when we culture cells in embryo bodies, the MSC subculturing has been shown to be much more efficient afterwards. There's also higher expression of anti-cancer factors in embryo bodies, and we can control the differentiation because we know that size and quantity of EBs affects differentiation, so we can control those parameters. So we have many biomaterials with which we can make certain scaffolds that we'll talk about later. These can be natural or synthetic. We have many different types of materials, either natural or synthetic. But synthetic ones are generally preferred because we can standardize the process much more. And natural biomaterials sometimes interact with cells in ways we don't want them to interact, right? And these mimic the biophysical and biochemical characteristics of SC niches, allowing for direct differentiation, right? Rather, to direct differentiation. So we have many different types of scaffolds, and there's types and advantages to each one of them. So one of them is prefabricated or rigid scaffolds, which generally require cells to be seated or migrated into the scaffold, and they're rigid and stiff generally. Whereas self-assembly scaffolds usually use cross-linking methods, which are often reversible by changes in pH, temperature, or other factors. And they're great for mimicking the natural soft tissue in which some cells grow. These scaffolds may incorporate growth factors or other chemical agents that might help to functionalize them. So certain challenges we can encounter here, for example, hydrogel scaffold-grown ESCs still require subculturing, and despite the fact that 95% of subcultured human pluripotent stem cells retain pluripotency after 50 passages, we do not want materials that require so many subculturing processes, right? Like we want to limit the amount of times that we need to subculture because, well, that's not good. So further consideration, we know that 3D scaffolds have shown improvement in MSC cell implantation outcome. We also know that we can control differentiation through mimicking certain in vivo aspects of in which cells generally are found in. And we also have to take into account that certain conditions might affect MSC viability, and these include cross-linking density, RGD concentration, and scaffold degradation, right? So we also have dynamic 3D culture. We know that bioreactors can provide advantages over 2D culturing, but we generally need well-defined temperature, pH, and dissolved oxygen parameters here. We also need to remove waste and metabolites from the culture. And constant steering is generally required in these kinds of cultures. So mechanical force bioreactors may also help mimic tissue and the physiology in which cells grow. So that's good if we want to control differentiation. Another type of spheroids are microcarriers, and these are small beads made of diverse materials like polystyrene, gelatin, dextrin, and collagen. And these can be optimized for expression and differentiation. These also help limit the effects of aggregation, the negative effects of aggregation that happens in embryo bodies, because the center of the spheroid doesn't necrotize, right? So these can be used for the further expansion and differentiation of ESCs. Microencapsulation techniques, unlike the previous one, the microcarriers, they aim to mobilize cells within a semi-permeable membrane, and this technique also protects against agglomeration and the necrotizing of tissue. The microenvironment can be modified to maintain stemness or to induce differentiation. Agarose and alginate are common materials for this purpose, and these can also be used with dynamic cultures, much like microcarriers, like we see in the picture here, right? So we finally have microfluidics, and these allow for studying the conditions similar to those in vivo. We can develop organ chips, kind of new technologies with microfluidics, and we can also study cell-secreted signals, right? So this is kind of a newer approach, newer kind of technology, but also very useful.
Going to present about the different challenge that isolation of stem cells may represent. So first we have the heterogeneity of culture cells, and it's related with the two-dimensional culture techniques because they are very expensive and they have a limited expansion of stem cells. So Also, the directed differentiation is partially achieved, so they have a lack functionality that it's important to study. And comparing with the three-dimensional models, we have a fail to achieve in vivo conditions that help the cell growth. Now we have the clonal growth, that a decrease in clonal growth represents a reduction of cell renewal capacity. So normally the subculturing of mesenchymal reduces the clonal growth and in consequence the self-renewal potential. So it's important to develop new methods to produce large-scale and long-term expansion of stem cells. Now we have the biomaterials related with the three-dimensional culture. The real challenge here is to identify appropriate biomaterials in order to develop suitable scaffolds to mimic the in vivo conditions for the stem cells, because these conditions are a mimic of biochemical and biophysical properties of the microenvironment. Now we have the optimization of culture conditions related with the standardized methods, because as I mentioned before, the two-dimensional culturing methods are commonly practiced but less efficient, and the three-dimensional culture system are more efficient, but they need more parameters like the biomaterials or conditions with the media. And now it has been studied that the dynamic bioreactors, because they have a better differentiation of cells comparing with the static bioreactors, but they can produce stress in the cells and have a negative impact in the
Another one
viability.
of the main challenges that these new technologies and methods are bringing is the functional activity. The challenge is that cells should have optimal viability and exhibit optimal activity in every culture method. And this is a problem sometimes when working with cells because this cannot be achieved as easily. Another one of the challenges is that culture cells should have an optimal activity, and this activity should be comparable to in vivo cells. Another one of the main challenges is that culture cells should be able to integrate into any tissue without any complications and with a high success rate. So, this is why it is very important that cell conditioning is considered before the use of the cells in the clinical purposes so they can integrate into the tissue without many problems. Future studies should investigate the temporal factors that emulate in vivo conditions and create microenvironments ideal for stem cell culture. Another one of the main challenges categories is the culture scale-up, because the current methods yield only millions of cells. Allogenic cell therapy for multi-center clinical trials require billions or trillions of cells, so it is not possible at the moment with the current methods. A scale-up of the required magnitude are not possible now. We require substantial advancements on the technologies and on the techniques. Also, large-scale culturing allows us to use the same cells on multiple patients, making the results more reliable. A problem for large-scale culturing is that it requires combined efforts from different areas. It needs researchers, it needs biotechnologists, it needs engineers, and they all need to be working together. So it is complicated and it is also very costly. Okay, now we're gonna talk about the conclusions. The conclusion of this topic is that the culture of stem cells has significantly advanced in the recent years, but it still has a long way to go. Most importantly, developments have been achieved in bioreactors and in the use of biomaterials. The stem cell culture using bioinductive, biodegradable, and biocompatible polymers help expansion, improve functionality, and help with the differentiation of the cells. Also, we've learned that the 3D methods solve the limitations of the 2D methods and can expand cells to billion folds. These methods provide the necessary for clinical trials, pharmaceutical applications, and biotechnology applications. And these methods will accelerate the therapeutic use of stem cells to treat diseases and disorders.